All right, everybody, welcome back. So here we are with part two of our discussion of lipids. Let me scroll down to the appropriate site where we left off. All right, so the second part of this chapter is primarily dealing with lipids and membranes, right? We talked about a lot of the different types of lipids that are biochemically important last time, and so, a lot of them we said they're present in membranes, right? Well, now we're going to see actually sort of how those actually present themselves, how they actually function while they're in these membranes. All right, so of course, as we've learned, lipids are pretty freaking important for membranes. They're a primary component of all common biological membranes, cell membrane, nuclear, mitochondrial, etc. And so, what are they there for? Well, some of it is basic protection, right? It's sort of like building the bricks of your fortress, right? The lipids, in a lot of ways, serve as the bricks. But also, they do have some other more specified functions, right? They also help to assist in transport. We have to get stuff in and out of the cell, and then in and across membranes, and so the lipids help us do that. So, let's take a look at how they are used. So, if we look at the cell membrane, for example, you guys have heard this ever since high school, right? What is the cell membrane made out of? It's made out of the lipid bilayer. And so, when we said bricks, they're literally sort of the bricks in the wall. You can sort of see that here, right? The lipid bilayer is the primary component of the cell membrane, and this is how it's arranged. Kind of similar to a micelle, right? You have a bunch of lipids, and they're kind of represented with the circles as polar head groups, and then the little squiggly lines as hydrophobic tails, right? And so anywhere there's water, that's where the polar head groups are going to point because they're hydrophilic, right? So it's a bilayer because there's essentially two layers of lipids. One of them, let's say the top layer here, has the polar head groups pointed outside of the cell, right? And as we know, human beings are composed of mostly water, so this is going to be an aqueous environment. On the other side, the second layer, the one on the bottom, the polar head groups are sort of pointed on the bottom, sort of towards the inside of the cell. The inside of the cell is an aqueous environment as well, right? So both on the inside and the outside of the cell, you have an aqueous environment. And in the middle here, hiding away from the water as best they can, are the hydrophobic tails, right? They're lined up with each other. And it sort of forms like a little like a little sandwich, right? The polar head groups are the bread, and then the hydrophobic tails or whatever you put on a sandwich, cheese, peanut butter, whatever. And if you look at the other photo there, B, you get sort of, I don't know if it's more of a 3D look at it, but you get more of a gauge for the sort of the depth and overall structure of these guys. All right, so let's take a look at some of the characteristics of this lipid bilayer. All right. It's a really simplistic analogy when I said sort of bricks in a wall, because unlike bricks in a wall, there's a lot of movement in and around the lipid bilayer, right? Those lipids are not just sitting there perfectly still. They're constantly moving around. So it experiences what we call a lot of fluidity. Right, they can move around. And it's not unusual for literally a lipid, whatever it may be, a phosphoacylglycerol, a fatty acid, whatever, to literally migrate back and forth to different places within the lipid bilayer. All right, so it's very fluid. However, you can see some common patterns. Number one, composition. There are many different types of lipids within the lipid bilayer. There's a couple of them that are probably seen more often than not. Phosphoacylglycerols. Yeah, here they are. Phosphoacylglycerols and glycolipids. They're probably the most common ones that are found. Although, like I said, you can see all sorts of them. The bigger, bulkier groups are most commonly found in the outer layer of the bilayer, the one that's where the polar head groups are sort of facing the external part, or they're facing the outside of the cell. And the idea is why this might be. Again, it's a very, very practical reason, right? The bigger, bulkier ones are going to take up more space. They're going to be more rigid, less fluid. And so, when you're assembling the outer wall, I guess you could say, they're going to take up more space. And that outer wall, excuse me, is going to, by nature, take up more space because it's covering a larger circumference. The total fluidity or rigidity of your bilayer depends entirely on the types of lipids that are present. And if you remember a lot of our discussion previously about the characteristics of lipids, especially fatty acids, right? Saturated fatty acids, unsaturated, trans fats, etc. A lot of that still applies, right? If you have a bunch of saturated fats, for example, in your lipid bilayer, again, those hydrophobic tails, they can kind of really stack up close to each other and make a denser, more rigid structure. Whereas if you have something with a lot of kinks, right, a lot of cis unsaturated portions, or other large groups that are sort of getting in the way, they can't get together as well, so they can crowd, and that makes things more fluid, less rigid. If you have a lot of cholesterol present, that makes it more rigid because cholesterol has that fused ring system. That fused ring system, again, is a system that can very much sterically stack upon each other. So you get stacking of aromatic rings, and that makes it much more dense and more rigid, right? So just sort of depends on what's present. This little picture right over here shows a snapshot, and it's kind of cool because, again, you have to look at the full photo of this, hopefully in your PowerPoint on a real computer or in the book, where you can see that pretty much all the types of lipids that we previously mentioned, they're here, just in differing amounts, and it's kind of cool to see them really how they line up like this. Some other general characteristics here. You see this in all membranes, no matter whether they're plants, whether they're animals, whatever, right? However, there are some differences. Animal membranes will tend to have higher percentages of saturated fats, whereas plant membranes will have higher percentages of unsaturated fats. Also, plants do not have cholesterol. That's a big difference. So in general, plant membranes are much more fluid than animal membranes because of this difference. Also, that plays into, excuse me, had to take a drink there. Again, that kind of plays into the whole plant-based diet being a little bit more heart healthy, right? Because by nature, when you're eating plant matter within their cells, within their lipid bilayers of their cells, they don't have as much of the quote-unquote bad stuff, right? The saturated fats, the cholesterol that animals do. So you're literally controlling the amount of those things that you get by consuming lipid bilayers. All right. So lipids, of course, are going to be in a lipid bilayer. Duh, that's just common sense, right? But it's not the only thing that's present. You got something else very important, and that's proteins. Every cell membrane, or every membrane, is going to have some proteins in it. And these will usually have very specific functions. There's a couple of different types that are commonly found. First type is called peripheral proteins. And if you think of the word peripheral, right, what does that mean? Well, it means sort of, at the edge, on the periphery, right? Not embedded. That's exactly what it means here. So these are really basically just sort of located on the surface of the lipid bilayer. They're sort of loosely associated. They're held by non-covalent forces. That means it's going to be held sort of stuck there by your hydrogen bonds, dipole-dipole, van der Waals, those type of interactions, those intermolecular forces. Integral proteins are the ones that are actually within the membrane. They're actually within the lipid bilayer. Within all those lipid bricks, you have these membrane or these integral proteins sort of wedged in there, and they are actually held covalently, right, through actual chemical bonds. The most common ways they are held are through myristoyl and palmitoyl groups. And so let me elaborate on this a little bit on the whiteboard. All right, so this is something that your book kind of glosses over a little bit. I'm going to go in a little bit more in depth. All right, so one way that these proteins are commonly held in, is through a process called and I'm going to butcher this because it's very tricky to say, S-palmitoylation. Ooh, I said that well. Congratulations to me. All right, S-palmitoylation. So, I think I put an extra Z in there at that time. S-palmitoyl. Okay, I should have stopped the first time. So, this process. All right, so this is based off, the name here is based off of this. Let me count my carbons real quick. 16. 1, 2. All right, so this is palmitic acid. Right? So what happens pretty commonly is that a protein will find a palmitic acid lipid, it's a fatty acid, present within your lipid bilayer. And what's going to happen is, I want to draw the little squiggle here. Squiggle means integral protein. All right, and so what will happen is one of these integral proteins will have a cysteine residue somewhere in it, right? And he knows cysteine residues, one of the important factors, or one of the important features, is that they have thiol groups. Okay, so what can happen here is these guys can link together through a thiol, through actually what we call a thioester linkage. Right? These two end groups, this carboxy group of your palmitic acid and the thiol group, can react. And you can form this little bridge. C double bond O with an S on it, that's referred to as a thioester. So S-palmitoylation basically is the linkage of palmitic acid to the cysteine residue of your protein through a thioester linkage. And it literally helps the integral protein stay in place within that membrane and be available to do whatever it needs to do. There's another type, major type, that's in here, and that is called S-myristoylation. And so the only difference here is that instead of using a palmitic acid residue, first of all, use a myristic acid residue. I'm counting my carbons, right? So palmitic acid had 16. Myristic acid has 14. So 2 fewer acid groups, and then it's going to anchor itself to the integral protein, not by a cysteine residue, but a glycine. Specifically, a glycine at the N terminus of your protein. All right, so, or the N-terminus of your glycine, sorry. Um, so basically, you're going to have the NH2 group of glycine reacting with the carboxylic acid group. Right there. And that forms your standard amide linkage or peptide bond. So this one's a little bit more regular, I guess, as far as basically forming a peptide bond, but in this case between an acid and the N-terminus of your glycine, whereas the other one formed basically a new group, a thioester. So, kind of cool. But those are the major ways in which these integral proteins are sort of wedged in and held in the middle of your lipid bilayer. Now, and you can see in this photo you have just some sort of simplistic examples. Basically all the ones in the middle, the yellow, 1, 2, and 4, those are integral, and then peripherals literally are sort of on the surface. And there you can see the two linkages that I just mentioned. All right, so we got integral proteins. They're sitting in the middle of the membranes. What do they do? Why are they important? Well, they basically act like gatekeepers, doorkeepers, right? Because cell has to get stuff in and stuff out of the cell has to move stuff in and out of itself on a regular basis, right? You got to get deliveries and then you got to send stuff out. Well, the integral proteins, they're basically the ones that help to control this, right? Also, these proteins, especially and this is more peripheral proteins. Peripheral proteins are especially important in making receptor sites, right? We've talked about this several times. Our cells, many of our cells have many different types of what we call receptors. This is where an external compound, whether it be another biochemical compound or something else, can come in and bind and either activate or deactivate some sort of process, right? That's commonly referred to as signaling in biochemistry. All right, so we got transport and we got reception here. So let's take a look at transport first. And you have heard these terms since forever, probably. You probably heard this in middle school or something. But we're going to talk about them a little more and see exactly how the proteins facilitate this. All right, couple of big broad categories of transport. You have passive transport and active transport. So passive transport first. That's when you have a substance moving along a concentration gradient. In other words, you're going from an area of great concentration to low concentration. All right, so let's say we had a boundary here. We have a bunch of X's. I'm going too fast, but I'm getting sloppy. But you get the idea. There's a bunch of them there. There's just a few over there, right? So passive transport would be literally the movement of X's from this side over to this side. Okay? The difference in concentrations is referred to as a concentration gradient. This is easy, right? In a case like that, it's energetically favorable to go from an area of high concentration to low concentration. And that's very much kind of a common sense explanation, right? If you had two rooms, one of them was absolutely packed with people and the other one only had like 2 or 3, wouldn't it make sense for some of these people in this crowded room to sort of migrate over and even out the numbers? It makes the overall situation more stable, and that's what happens here. It's energetically, thermodynamically more favorable for these species, whether they're ions, molecules, whatever, to migrate and even out the concentrations a bit. All right. So the most common way of doing this is through simple diffusion. All right. That's where it just basically does its thing without any major assistance. And on a normal basis, that happens all the time within a cell, especially with smaller species, small ions, small molecules like maybe water or oxygen or carbon dioxide or something like that, right? However, sometimes you have to have a little bit of help. Not in the form of energy though, right? It's more like you have something that you have a concentration gradient in the molecules, the species. They want to even themselves out concentration wise, but they literally don't have an avenue to get there, right? Maybe they're too big to get from one side or the other. And you see a lot of examples with these. Glucose, right, is a classic example. Glucose is a simple sugar, but it's going to be bigger than your small molecules. A number of ions, right, especially the larger ions, they have trouble moving through. So this is where some of the integral proteins can become important, right? If you have and again, I'm drawing little cartoons here. This is dramatically simplified, but let's say I should have drawn holes in the last one. I'm going to draw holes here to demonstrate where the species might be able to come through. Right, so for something like, an O2, might be easy for it to squeeze through, right? Whereas if you have something like glucose, and I'm just going to write the word glucose because I don't remember the structure of glucose offhand and I don't want to look it up. But glucose, too big to fit through one of these little doorways, right? But if you have a spot where there is one of these integral proteins maybe embedded within here, almost acts as like a little tunnel that can get through, right? So in essence, during facilitated diffusion, these integral proteins basically kind of act as tunnels. Now that's simplifying it a little bit, but that's essentially what happens, right? It's important for bigger molecules and for certain types of ions as well. Get in and out. Since this is still going along a concentration gradient, you're still going from an area of higher concentration to lower concentration, this is not giving the system energy. It doesn't need energy to drive the process. It's still a spontaneous process, but it's just giving you sort of a physical means to be able to do it. All right. Now, the cases where you actually need energy, that's going to be a different classification. That's going to be called active transport. All right, and that's where you're moving against the concentration gradient. So, I'm going to draw a dotted line instead of the solid line. That might be better. Right? So, that would just be just the opposite of what we were talking about before. Right? That's where maybe you would have this situation and you would actually have maybe a couple of these trying to get into the crowded room, right? That's not thermodynamically stable. That's not a spontaneous process, right? So it's not going to happen by itself. You're going to have an energy source to help drive that, all right? And so there's a couple of different ways that you can get this energy source. The first one is from a direct energy source, like ATP, right? If you cleave a phosphate group off of ATP, that is thermodynamically spontaneous. It releases energy. And that energy that's released can be coupled to diffusion against a concentration gradient, right? That is an example of what we call primary active transport. Again, there's several different examples of this. Hold on, my computer went to sleep. I'm using this. I've got like a big setup here, so I'm using like multiple things as source material. So I got to wake my computer up so I can see my PowerPoint on the real computer. There we go. Okay. So there's several examples of this. A good classic example again is our old friend, the sodium-potassium pump. So we mentioned this, previously when we were talking about how modification of proteins can change their conformation, right? And we talked about phosphorylation and dephosphorylation of, of the enzyme ATPase, which is embedded. ATPase is an embedded integral protein within the lipid bilayer. And so what's the function of ATPase? Basically, it functions to... Rewind. Got tongue-tied a little bit there. The function of this ATPase is to drive concentrations of sodium ion and potassium ion to certain levels inside and outside the cell, right? It has to create enough, there's basically a certain charge distribution that has to be maintained to maintain something called a resting potential. And this is something you probably went over in physiology, and I'm not going to go over it because of that, and also because that's not the purpose of this discussion. The purpose is to see what actually happens to this integral protein. So this one's a little bit more of an involved diagram than the one that you saw before, but in a lot of ways it kind of shows the same thing, right? If you look at the very top of this circle, you've got your lipid bilayer, and that thing that sort of looks like a tooth, that's your sodium-potassium ATPase that's embedded within. All right? And so, again, its function here is to move sodium and potassium where they need to be. All right, so let's say this is the inside of the cell, and this is the outside of the cell. So, on each side you have a bunch of sodium and potassium ions. So, let's say got a bunch of sodiums here, and you got some potassiums. And on the other side, you got some sodiums, and you got a bunch of potassiums. Whoops, why did I write Ka? That's not right. There we go. Getting sloppy here, sorry about that. Okay, so basically what's happening is you can see there's a concentration gradient for both sodium and potassium, right? On the outside of the cell, you have a higher concentration of sodium than you do on the inside. Conversely, you have a higher concentration of potassium than you do on the outside. However, what sodium-potassium pump is trying to do is it's trying to take 3 of these sodiums and pump them out to the outside. Potassium is also trying to take a couple of potassium ions and pump them to the inside. Both of these are against concentration gradients, so you got to have some energy to drive them, right? And that's where the sodium-potassium ATPase comes in. All right, so if you look at the top of the circle there, basically what happens here in that first step is that 3 sodium ions from the inside can bind to your ATPase, and at the same time there is an ATP that gets cleaved, right, where you have a phosphate group that's chopped off, and that's attached to your ATPase. So there's actually 2 important things that happen here. Number one, you're cleaving that phosphate group from ATP. That gives you energy. That gives you the energy you need to drive against that concentration gradient. The other important thing, and this is the one that we went over in a previous chapter, is that when that ATPase is phosphorylated, it changes its conformation. And so you can see the little tooth shape is actually inverted now, so that the roots of the tooth are pointed on the outside instead of the inside. And that allows those sodiums to be sort of dropped off on the outside. Okay? While it's in that shape, then it happens to pick up 2 potassiums from the outside. You get a dephosphorylation of that enzyme through hydrolysis with H2O, and it changes its conformation back to its original tooth shape with the roots pointing inside, allowing the potassiums to be dumped off. All the while, the energy that was originally released from that ATP helps to drive against both of these ratings. Then your ATPase is back to its original position, lather, rinse, repeat. Right? So, certainly not the only example of a biochemical process that uses ATP to help drive it, but a really good classic example. Actually, no, I need to be here. Sorry. All right, let's go back up here. Third bullet point. There is another type that there are other ways that you can get energy other than using a direct energy source such as ATP, right? And this is where we get what we call secondary active transport. So you're still going against the concentration gradient, but you're getting your energy in a different way. And usually what this means is you have some other type of process that's going on that's it's not the same as cleaving a phosphate off of an ATP. It's not necessarily a direct covalent reaction, but some other favorable physical process that it can sort of piggyback on. I think giving an example here is probably the best example. Frequently, within biochemical systems, you can see something called a proton pump. And if we were to get to electron transport, you would see this. I don't know that we'll get there or not. We'll see. A proton pump can help drive things. So here's the good example here. So basically what we're trying to do here is we're trying to get lactose from the outside of the cell to the inside of the cell. That is not a favorable process because the concentration of lactose is going to be higher outside of the cell, higher inside of the cell than on the outside. So naturally, lactose has a hard time moving inside the cell because of the unfavorable concentration gradient. Right? So that's bad. But guess what? That same integral protein, that same integral enzyme protein, galactoside permease, is also able to allow protons to come in. Right? And if you look at the concentration of protons outside and inside of the cell, there's a favorable gradient, right? There's more protons on the outside than on the inside, so having protons come in, that's a favorable process. So, in essence, when these protons come in through galactoside permease, that creates enough of an energetically favorable situation where lactose can kind of piggyback on it and say, hey, I'm going to hitch a ride here and come through and use some of your energy to get me across. And that's exactly what happens, right? So they can both move through the same tunnel, all right? So why do we call it a proton pump? We call it a proton pump because eventually this happens enough, our proton concentrations are not going to be where we want, right? If we keep doing this, then we're actually going to have a reverse gradient where we're going to have more protons on the inside than the outside, and that's not good. So within a lot of membranes, you have this proton pump system. Basically, it's a series of different processes, reactions, etc., that allow protons to be shuttled back to the outside. How those are set up depends on the situation. There's actually lots of different ways these are set up, but it gives you a mechanism for reforming this positive gradient that allows, in this case, lactose to sort of hitch your ride. Yeah. So again, it's all about it sort of hijacking energy sources from somewhere else. In this case, it's not a chemical reaction, but from a favorable movement along a favorable gradient. All right, so that's sort of, in a nutshell, how integral proteins are used in transports across cellular membranes. Again, as we get to some specific situations in later chapters, we can see exactly how some of these work on a closer up basis. All right, so the second major function here of these proteins was as membrane receptors. And again, these are more going to be your peripheral proteins, although some integral proteins can be involved, but peripheral proteins have more of a role here. And so it's very much like we were talking about when you form an active site of an enzyme, right? So one of these proteins can be folded up in a certain conformation, and when it does so, you create a certain pocket that has a place where some sort of molecule can come in and fit, right? It can fit in there like a little puzzle piece, and when it does so, that often serves as a signal to maybe start or stop a particular set of biochemical reactions or processes, right? Just like enzymes, these can be inhibited by various things. These can be with other binding sites that actually bind inhibitors. This can be with analogs that actually act as competitive inhibitors, and it also can be related to concentrations in a feedback mechanism loop like we saw with ATCase in a previous chapter. I'm going to show you an example of one of those here, and this one that you've probably heard of. It's by using is the uptake of cholesterol through what we call low-density lipoproteins, or LDLs. If you've ever had a cholesterol test, you may have heard of the terms HDL and LDL. These refer to lipoproteins. Lipoproteins are basically these big balls of that's composed of both lipids and certain types of proteins. Low density ones are not packed together. Oh, excuse me. Sorry, I had hiccups. The low density ones are basically, they're not packed together very tight, so they're kind of puffed out. They take up a lot of space. High density ones are able to pack up and form smaller structures. It's the low density ones that are generally bad for you. Anytime you have a cholesterol test, you have what they call it good cholesterol and bad cholesterol. It's actually not cholesterol they're referring to. It's referring to the concentrations of these lipoproteins. If we were to get, and I don't think we will, if we were to get into the lipid metabolism chapter, we would go over this in more detail. But for now, we're going to look at this little cartoon here, this little structure. I like this. If you like the game of life, whenever you have a kid and the game of life, you have to get one of those little pegs and put it in the car. This is what this reminds me of right here. But this also shows a good example of how LDLs work in the body. So on a cell, in this little diagram here, you have a bunch of these little green discs with holes in them. These are the peripheral proteins that are forming your LDL receptors, right? LDLs, they have a landing site. They don't just float around aimlessly. They are taken up by the cell, right? Because the cell needs what the LDL is carrying, and that's usually cholesterol. All right? So you have a bunch of these receptor sites, and these little pink game of life pegs, these represent the LDLs. And so what happens is, as LDLs come in, they bind to these receptors. And you can see that over here on sort of the left-hand side of the diagram. All right, so what happens is when they bind to a receptor, that sends a signal to the cell that, hey, there's an LDL here. It's bringing a delivery of cholesterol. Let's bring it in. I want to get that cholesterol. So what happens is you will have a process called endocytosis, where the cell will sort of bring in that LDL receptor complex and sort of pinch it off. You've seen this in biology classes with other things. This one's specific to LDLs. All right, so it pinches off, and it brings in your LDL receptor hybrid or complex, and then it'll break them apart. It'll take off the LDL, and it'll recycle the receptor and push it back out to the outside so it can bind to another LDL. Lather, rinse, repeat, right? Then it takes the LDL and basically kind of disassembles it, and in the process takes away the cholesterol for use, however it might need it. All right? So, why are LDLs bad for you? Well, if you have too much cholesterol in your cell says, I don't need any more cholesterol. Please, no more cholesterol. So it'll take in an LDL receptor complex. It'll disassemble it. It'll get the cholesterol. The cholesterol itself, if it can't be used, will actually act as an inhibitor for the synthesis of new LDL receptors. So in a nutshell, too much cholesterol means these LDL receptors are either not being recycled to put on the outside, or you're not making new ones. So that means cell has fewer receptors. The cells have fewer receptors. The LDLs, they have nowhere to go. They're stuck within the bloodstream. And eventually you get a buildup of all these in your bloodstream, too much that you shouldn't have, or that you aren't meant to be in there. They can pile up and form blockages. Right? So, if you're looking at why cholesterol is bad, it's a multi-layered process. Number one, if you have too much of it, these LDL receptors are not produced and the LDLs have nowhere to go. And then second of all, within the LDLs, right, the LDLs have high concentrations of cholesterol, which have structures that really lend themselves to forming blockages. So, bad on multiple levels. HDLs, you don't have this problem. So, that's why the HDLs are the quote-unquote good cholesterol. All right, so we're going to shift gears a little bit here. No discussion of lipids is complete without looking at vitamins. A number of the vitamins that are important to our everyday functions are in fact lipid or lipid-based. All right, there's basically a couple of different types of vitamins depending on their solubility. Right, first group is water-soluble vitamins. All right, and that's self-explanatory. These dissolve in water. Vitamin C is a water-soluble vitamin. Vitamin B complex is water-soluble, right? We also have what we call the fat-soluble or lipid-soluble vitamins. These are going to be vitamins A, D, E, and K. If you ever forget, just remember ADEC. That will tell you the lipid-soluble vitamins. Why are they lipid-soluble? Well, why are any of these other lipids lipid-soluble? Or I guess that's self-explanatory. Why are any of these not water-soluble? Same common thing, right? They all have highly hydrophobic hydrocarbon tails at some point within their structure, right? And that's what happens with these. These lipid-soluble vitamins have big hydrophobic regions. A lot of them have conjugated double bonds. Ooh, do we remember something here from organic chemistry? Right, conjugated double bonds. In case you don't remember what conjugated double bonds are, that's where you have sort of these alternating double single systems, right? And we saw in organic chemistry that conjugated double bond systems, they can do specific things, right? There are specific reactions they undergo. This affects the wavelength of light that can be absorbed, etc., right? And so having these conjugated double bond sections gives these vitamins some specific functions. So let's take a look at a few of them. Let's start with vitamin A. Vitamin A, also known as retinol. Vitamin A is very important for vision, for sight. Okay? And so, it's not the only thing that's involved in sight, certainly, but it's part of it. It's especially important for our night vision. People that have retinol deficiencies oftentimes will have poor night vision. All right, so let's take a look at some of the chemistry here. So, vitamin A is derived from a compound called beta-carotene, and you've probably heard of beta-carotene before. It's a very common nutritional phrase, and also I used beta-carotene as an example of conjugation in OChem. It's colored, right? If you look at the structure here, this top structure is beta-carotene, really extensive conjugated double bond structure. And if you remember from OChem, we said that the more conjugated a system was, the higher wavelength of light absorbed. And so most organic compounds are, clear or colorless because they don't absorb or transmit within the visible region of light. Well, when you have conjugation, it shifts that transmission and absorption into the visible region, right? So beta-carotene will usually appear sort of an orange color because it transmits orange wavelength light. And we actually talked about how people can it's possible for people to consume so many carrots, which have lots of beta-carotene, that they can actually develop an orange hue. So I'm getting a little off topic here, but just kind of trying to remind you what beta-carotene is. All right, beta-carotene can be cleaved enzymatically. It can be sliced in two pieces. And when you slice it like this in the second structure, it forms this. This is retinol. This is the structure of vitamin A. It's still a highly conjugated structure, right? It's just shorter than beta-carotene. All right. So there's a reaction that happens within your eyes. There's a couple of reactions. All right. The first one is the reaction of retinol with an enzyme called retinol dehydrogenase. And basically what that does is, you can see in red, there's shaded an alcohol group on the very end. It converts that group into an aldehyde. All right. And by default, you'll see we have a bunch of double bonds in here, right? As you guys remember from OChem, hopefully, you can have different arrangements around double bonds. Remember cis and trans, etc. So you can have different stereoisomers. The one that's dominant here is this one that's highlighted as 11-trans-retinal. Retinal is what retinol is called once you convert that OH into an aldehyde. All right. So this is the dominant stereoisomer, 11-trans-retinal. That means that you have a trans arrangement around the double bond between carbons 11 and 12. All right, that's important because this is a biologically inactive form of retinol. This part plays no role in vision. However, there's an enzyme called retinal isomerase that basically can convert the 11-trans-retinal into 11-cis-retinal. So it converts that stereochemistry around carbons 11 and 12 to a cis instead of a trans. This one is biologically active. And so, once you form 11-cis-retinal, it can bind with a protein that's present in your eye. It's called opsin. And so basically what they did there was they drew part of opsin there to highlight the NH3 group that's present on its side chain. That NH3 group is going to react with the aldehyde group that we made, and it forms what we call an imine linkage. So basically